setup here.
Yeah, we had a good conversation. And
Actually, it's whether people
we had a very positive discussion about what's going on now, but I'm not counting my chickens.
Well, Sana went to open the door
Sad to
to
hear what
Daphne,
happened
so probably
there.
can also let other people in.
For everybody, because it looked like.
I thought there was genuine interest.
Yeah, and I even sent her a follow-up email trying to probe more deeply about it. It seemed like kind of a bit of a made-up reason. But her response was not any much more illuminating. You know, I was thinking that Harvard decided to give her a position in biology. But I don't know what impression that was.
So it's really hard for the seminars.
I don't really
that
know.
work
You never
in
know
this
these
building,
things.
they can open the door with the access
Anyway,
card,
this is
but
why
the
we
people who are coming from other departments, they have a hard time
Very good and they don't know it all.
getting through the.
So I'm optimistic about stuff.
Yeah.
Okay.
But then this door
Do we have any,
or this
are you still doing two or one?
Yeah, we're also looking at it.
What's the door open?
Yeah.
From Erica.
Well, we can wait a couple of minutes.
Bowers Lab at Penn works on autophagy.
Are we very strict on the timeline?
So she works with.
Okay, maybe I can start with the introduction while a few more people trickle in. So it's a very special day for me because I'm introducing not just a regular seminar speaker, but the person I work with for many, many years, my real scientific mentor of many years. I did grad school in his lab, a postdoctoral fellowship, and he really shaped the way in which I'm doing science now. So it's my great honor to introduce Dr. Geoff Rosenfeld, who is a distinguished professor of medicine at UCSD. Honestly, I could go on for hours talking about all the things he has done in life and all the awards and the papers he has published. I think we are now to the point that they are in the 500s counts. The award he got, I can just say he is a member of the National Academy. Has been for a long time. He has been an HHMI investigator for almost 30 years, just stepped down. Really, he has left an incredible mark on the field of transcription from the beginning, from recording messenger RNA and understanding how to do in vitro transcription and translation. And what I've always been very impressed of is the fact that he has moved farther ahead of time. So when people were not thinking yet about genome-wide, he was already thinking about that. When people were not thinking about an answer, he was already thinking about that. And to be honest, that was stressful in the lab because to keep up with what he was talking about when we had no idea yet where the field was going was a challenge, but it was also very exciting. And so I won't spend time telling you about all his achievement. I think most of you know some of his work if you work in the field of transcription. I just want to mention that maybe one of the most important legacy that he leaves to the field is all the trainees that have spun out of his lab and are really now PIs all across the world, from UCSD to other places in California, UCSF, to New York. He was there in the last couple of days, Columbia, NYU, here. All the way to the opposite side of the world, China, Japan, going through Europe, Karolinska Institute, everyone. And so I think we all met together a few years ago, and it was a very special time. And I'm really happy to have the opportunity to share with you some of his great new science. I'm sure it will be an exciting seminar. And I would also encourage students and trainees who will join for lunch, ask him about all the things that happened in past years, because he's really an historical memory for how transcription has developed through the years, and he has some really cool story to share.
Thank you. Okay, let's take a second to just get this put away. Okay. Well, good morning, and really very special to actually have this invitation from Valentina, because as she said, many people have gone through the lab, and I think that's we're almost up to 70 people who've gotten jobs elsewhere. But Valentina is very special, one of the most fun times I've ever had in the lab, and it's great to see what she's gone on to do. Now, I feel a little bit like a stick in the mud, because Valentina's gone on to do new things, metabolism. Here I am, still doing transcription and transcriptional mechanisms. So I guess today I tried to make a little bit of an enigmatic title. So the whole thing is, of course, in every cell, there's so many regulatory signals, a universe of signals, and you have to integrate this into a final, very precise transcriptional pattern and profile. How do you do this? And so, of course, therefore, to put it into a simple perspective, the endocrine system, the paracrine system, all intercellular signaling systems have to regulate these fast homeostatic programs, and they have to be very, very integrated and very precise. So, of course, a big advance was the realization that almost all of these signaling systems are not uniquely, but operate through enhancers, and that the binding sites for the transcription factors that mediate so many of these signals, like ligands and cell surface pathways, factors, work through enhancers. And so it's really remarkable to me. It's 44 years ago, Walter Schaffner in Zurich was responsible for elucidating what actually has, I think, been not as recognized as it should be for such a monumental finding, that there were elements in the genome that were very distant from the actual initiation site of the promoter that had nevertheless a very profound effect on gene transcription. And others later confirmed this, obviously, but the initial regular, fervor in the field was this was going to how you do development. This was turned out, I think, that there were animal models that were done, and so these were the developmental regulators. But what emerged over time is that actually these very enhancers that were so important for turning a gene on in a specific cell type were the same elements that let signaling systems regulate the gene. And so that understanding, of course, makes great sense. The same way you get cell type determination, you use this later to have signals perturb the system in a very specific and profound way. So then these large programs, how do they work? And that was the connection, that it was the enhancers were the major binding sites for these signaling systems, not promoters, where, of course, all transcriptional initial biochemical work had focused. Of course, there was the transcriptional units, they had the coding genes at that time, and non-coding genes as well. But actually, the program started from signaling at the enhancer. So that, if that's true, then you think that the enhancer activation must occur temporally before promoter activation, which is exactly what happens. So the enhancer activation temporarily precedes, this happens to be an example of the estrogen receptor, and and if you start doing the time, it's actually measurable and clear that the enhancer is the first activation. So in other words, if we want to say what is the most important transcription unit for the signaling system, it's the enhancer as a transcription unit, which then has to give its okay for promoter to then take over and give us a coding target gene or a non-coding gene. And of course, then I won't go through this. From the early work in bacteria, everybody, of course, assumed, but now it's not as simple as this, that the promoter must communicate with the enhancers. That they use the term looping, overuse the word looping, we'll call it looping for now. And So the conclusion that the first part, the most simple thing, is that signal and ligand band enhancers are temporarily activated prior to their cognate target coding genes. So in the case of an example based by the nuclear receptor, the estrogen receptor, or any nuclear receptor that's ligand dependent, of course the enhancer is established during the cell type determination. It's open, it already has cohesin associated, it already has histone marks, and nevertheless it is the signal, in this case ligand, estradiol 17 beta, that allows the estrogen receptor, and it's more complicated as I'll get to bind to the enhancer and to then start the program, not really markedly increasing the amount of cohesin that's there, but definitely increasing to some extent, and in the case of estrogen receptor, actually allows some condensins to bind, and now we've activated the target gene. So that scenario seems to recap for almost every signaling pathway. So the next little fact that I thought was very interesting by Jason Liu was the idea that's now called in the literature as promoter hotspots and enhancer hotspots. So the idea is that estrogen receptor binds, and of course it should be very simple. It should activate the enhancer. What do we mean by activate the enhancer? Well, in the most of the cases, estrogen bindings, depending on the lab that's done it, we all get between 30 and 40,000 estrogen bindings. It's a lot of sites, and in the target tissues for estrogen, or for most nuclear receptors, they're very profound programs. They really are the big driver in their target tissues. So if that's the case, then how many actually link to genes that we know? Well, above the enhancers that really are strongly activated, let's say the average is 1 or 2,000, and another 2,000 to 3,000 moderately activated. So out of the 40,000 sites, only a fraction really seem to have a lot of functional significance. So then what else is needed? And the observation that was so surprising is that on a lot of these enhancers, in fact, these strong sites, they were not the only thing that were bound. And he showed that a number of things were essentially assembled at the site on DNA. So it's not a pre-existing complex, but it brings in these other proteins. Well, these are really strange bedfellows, because here's GATA3, RAR alpha and gamma, DNA binding transcription factors, for which there were no obvious sites on these enhancers. And not only that, GenFOS, AP2 gamma, STAT1, other things, and a very famous kinase, DNA protein kinase, catalytic subunit, that all came together in a 1 to 2 megadalton complex, which we call the megatrans. And he was meticulously mutated the DNA binding, very precise mutations that eliminated their ability of each of these factors to bind DNA, and they still just work fine. Except, of course, for estrogen receptor, which need to have its recognition element. So therefore, these were a assembled complex, and I'm not going to spend too much time on this point, but they were quantitatively very important. These seem to be the most prevalent. At that time, there was no way to do more than a 2-step chip assay, so we can only say that at least some of them were present at the same time. There's no way to know if they're all present at the same time, although there's a 1 to 2 megadalton complex that would include all of these and other factors I'm not showing in the complex. So that's the next thing, that for some reason you need not just the DNA binding transcription factor that's signal dependent, but others that normally would be signal dependent, but actually are not binding to DNA at all, but are coming as a complex. And it even brings condensins to this. I'm not going to go into that today. Okay. Now, I won't go through this detail, but of course,
there's been this craze, and deserved craze, for phase-separated material. It got attacked by Tijan. He says, look, complexes form just fine. You don't need condensate. There's obviously structures in the cytoplasm and nucleus that have every feature that would argue these are really, in fact, protein, these are phase-separated structures. Without getting into the argument, I think most people will agree these clearly exist, and there's no reason to think they can't exist on small structures like the enhancer. But everyone agrees with the statement that it has not been formally proven in the cell situation. So I will just call them condensates for the sake of the argument, but in my mind, all the arguments Brangwyn and others have made, they're completely valid and really should be eventually rewarded with some great recognition. Okay. These, as I'll get to today, what's really important about the condensates is that there are really many states of the condensate, and the properties that change in the condensate is really a key determinant of the details of the activation events. So what I'm going to do today is just arbitrarily, instead of doing the right thing, which is to give a detail about one story, I'm going to do less detail and try to give you what, after doing this, I guess our first paper on this was in 2013, so it's a decade of trying to understand how this works. In other words, this is the title of the talk, because we're saying, how do you go from an inactive to an active enhancer? And I think of it as leaping the Minotaur, so I'll explain in a second. So what I'm going to tell you about is that after you bind to your cognate enhancer, there are a series of, to say 5 phases is silly, that's what I mean, that's what we know now. It's just arbitrarily divided into phases. So what I'm going to try and tell you today is that the first thing you have to do is note that you have to activate the enhancer, which was previously generally the least active of all the enhancers. The stronger the enhancer is, for the estrogen effect, the less basal activity it appears to have. So how do you do that? And I'll try and tell you the story of how we are, since this is an incomplete story, how we think that happens. And then we go to another phase that is a story that's now published, which is, I find, quite remarkable, that in order to activate the enhancer, you have to do a form of DNA damage. Now, this is for me a sore subject that maybe Valentina remembers, but we had a finding in the lab that took us a long time to even get the courage to even submit. It was the idea that in order to transcribe effectively, it was required to get double-stranded breaks. When that paper finally went in for review, the reviewer said, as you could predict, this is absolutely ridiculous, because we all know that if there's a single DNA double-stranded break in a cell, the cell will die. And that was what all the textbooks said. Of course, we now know how laughable that is. The cell doesn't die. It's not just a question of that. The actual DNA damage is required for complex transcriptional programs. If you don't break the DNA, you will not have effective complex transcriptional programs. So that is an example of how a dogma gets so set that no one can even dare say anything else. So we took terrific grief. I took terrific grief at any place I gave a seminar that was laughed at, but not for long. And later, the first person to reproduce it was Li Wei Sai at Harvard, and in another system, in a neuronal system. And then, of course, now, everybody says, well, of course, if you don't transcribe and have breaks, you know learning and memory, for example. And so learning and memory requires damage, and now it's gone on to, well, that's why we sleep. That was for a long time. Why do you have to have sleep? It was to break down neuronal connections, so that everybody had said. I remember back in the days when down this lab, Francis Crick gave a talk on this, and he said, well, this we have to break down so we're ready for new neuronal connections, which is all true. But actually, why we sleep is to repair the DNA damage. There's a beautiful series of papers very recently, including one in zebrafish that I thought was very clever. And they used the simplest assay, which is gamma H2AX foci, and they showed that a zebrafish sleeps about the same cycle we do and gets that by that crude assay, they get DNA damage. And if they get 2 hours sleep, they get some of it repaired. They get 6 hours sleep, then they get completely repaired. So in other words, sleep is the opportunity to repair the damage of all your activities and thinking of the day before. So, okay, so now it turns out that another DNA damage system, the topoisomerase 1, which, remember, makes a single-strand nick in DNA and makes a covalent intermediate with the DNA, which is called TOP1cc, covalent complex. It turns out that is a required first step for the activation of not only the estrogen-dependent enhancers, but many other signaling enhancers, including for signals of inflammation and signalings in depolarization in neurons. So in other words, this is a very surprising thing. You use a system, again, so in other words, DNA, we have a field called DNA damage repair and transcription. Everyone has failed to appreciate for so long that, of course, DNA damage and repair is required for effective transcriptional programs, but better to survive more than a brief period on this earth, you have to have a very effective repair mechanism. And in the aging field, people always say, well, you know, it's really interesting, amongst mammals, we're some of the best, not the best. Whales are better, others are better, but we do some of the best repair. Why are we better? Why do we repair better than others? And of course, we've had to be able to get the more complex transcriptional programs we need and to survive long enough to use them to some effect. Okay, so then the next phase is the one I've told you about, the megatrans complex, that transcondensate. And then I'll tell you about a checkpoint release point. And finally, so what I'm going to talk about here, and the last point are unpublished data, and mostly complete, but not complete. Okay, so I gave the title, oops, we didn't quite all of that, but I got the title was that we're jumping the Minotaur. So for those of you who are not immediately conversant on Greek mythology and Greek lore, this is the fact that the Minos, King Minos, the Minoan culture in Crete, was really had his son Androgenos was killed by the Athenians, and he was so upset that he beat the Athenians and made them send 7 male men and young maidens every year to be devoured by the Minotaur. Who's the Minotaur? So the Minotaur is a classic Greek mythology story. Poseidon gave a very beautiful snow white bull to Minos for sacrifice, but Minos found it so beautiful, instead of sacrificing, he kept it alive, and of course, his wife, Pasiphae, fell in love with the bull and had a child by the bull called the Minotaur, half bull head and man in a body, but a very carnivorous type of beast, supposedly. And so he would presumably devour, so in order to maintain this, he had his architect, Daedalus, whose son, the famous Icarus, build a maze in the bottom of the palace. And this maze was so Minotaur could not escape. And at the third year of this, the Athenians sent, as one of the so-called volunteers, a warrior, Theseus, who agreed to go and with the daughter aminos, Pasiphae. She gave him the cord so he got into the maze and he could find his way back out. Anyway, he kills the Minotaur supposedly and ends the adventure of this event every year. Now, actually, this is the memories in Athenian pottery of the killing of the Minotaur. But this is the picture of the murals that are in Knossos that actually show what really was going on. Presumably, these Athenian hostages learned to do an amazing beginning of the bullfight. They would essentially grab the horns of the charging bull and essentially leap the thing and end up as on the other side, presumably alive. Probably a very high risk type of thing, and probably that was the attrition of the 7 maids and 7 males who came every year. But this is obviously was a beloved sport in Minoan culture. Okay, so basically that is the story of that, how we're going to go thinking about how we leap the Minotaur to activate an enhancer. So the first thing I have to tell you, of course, every signal, I don't have to tell any of you this, you don't just turn on genes, you turn genes on and you turn them off, and they're both very large programs. And the first thing is Yuliang Tang, I thought, did a really wonderful story when he took on the genes that went down. Why did they go down? Where did estrogen bind? Did it bind to the enhancers there too? It did. Well, so how come it went down? Well, the reason was obvious, because they were already very basally active. And when estrogen receptor was, quote, bound to those enhancers, they basically turned back off. They were repressed. And why were they repressed? And since it's a published paper, I'll just say that, well, it turned out that they were no binding sites for estrogen receptor on these large program of repressed enhancers. Well, so what happened? It was apparently they were indirectly brought into a condensate of some sort. And in trans, interestingly, though they could, for the first few minutes, actually have activation begin, then they turned into repressor, and the answer was, he solved very clearly, was a very interesting series of events that led to the degradation of the polymerase and the clearance of that enhancer. So that was, to me, a very interesting aspect of how trans recruitment of a DNA binding transcription factor can have a profound transcriptional program effect without ever having a site, and I'm overstating, for that regulating transcription factor. Okay, so then I'm going to tell his story that of this TOP1 nicking. So what he really said in this paper, that there was a code in metazoans for signaling pathways to need to make the single-stranded nick and make a covalent intermediate of a topoisomerase 1 in order to activate transcription. And first he did it in estrogen receptor, but I just want to tell you that he then went on with both by our behest and the reviewers' behest to see how broad this was. It worked for the androgen receptor, it worked for the NF-kappa B signaling program, it was the same for depolarization of neurons. So in other words, you really needed this event. And that was, since it's a published paper, I'm just going to summarize it as here. So that's our basally inactive or lowly active. It's an open enhancer. It already has been decorated with the right histone marks. It's already been decorated with cohesin, but it's when the estrogen receptor binds, it causes the activation of topoisomerase 1, and that makes this covalent intermediate with DNA. Now, that's interesting, and of course, as you all know, topoisomerase 1 has the property of only having ligase function, so you can religate the DNA, but you do need other help to make it more effective. But his big finding was, why in the world does that is that needed? And it's very surprising, and it took a year and a half for him to go from this first observation to the observation that there was a protein that recognized not TOP1 itself, but TOP1 when covalently linked DNA, and that other protein was, of all things, one of the most important partners of DNA damage repair, and that was Cu70. The Cu70 usually exists as a Cu70, Cu80 heterodimer, but in this case, it really just seems to be Cu70. And who cares? I mean, that's so surprising, yet without Cu70, you don't get activation. And then what does Cu70 then do? And there was quite a series of events that allowed him to do this. He realized that Cu70 itself, and we got a little fly from fly biochemistry, who had already noticed that Cu70 could interact with a heterochromin protein in flies. And so we checked, and sure enough, Cu70 could interact with one of them, which is called HP1 gamma. HP1 gamma is described as a very important repressor. But then there were two papers that appeared, and you can imagine, since they were heresy at the time, they didn't get into major journals. They claimed, oh, but sometimes it's an activator, but we don't know why. And the reason why it turned out to be because it gets phosphorylated. And if it's phosphorylated, well, why did it get phosphorylated? It got phosphorylated because estrogen receptor brings with it, as I told you, that DNA protein catalytic kinase, and this is a substrate for that. So now you recruit not just this repressive form, but this activating form of heterochromin protein 1. And then, again, thank you for other model organisms who had known, it was known that they could interact with something which was the certain ortholog of mediator subunit at 26. And you needed all of this machinery in order to increase essentially allow elongation of the eRNA, and apparently, although I realize that may not be the only reason, activate the promoter. So I thought this was, to me, a really quite remarkable story, and yet another example of why DNA damage and damage repair is so integral to our transcriptional program life. So activation of TOP1 by signal-dependent transcript factors appears to be required for most all signal banding enhancers, and these are the ones we've actually tested it on. All, I have no idea, but I would like to say many or most. Okay, now, that brings up the next question. Why the strong enhancers are strong, and why are weak enhancers weak? And that, of course, brings us to a question that everyone loves to investigate now. There's probably a million grants on this, a million papers on this, which has now been, and I give Mike Levine credit as usual for being a pioneer in the vocabulary. Why are these grammar of these enhancers that makes them strong or weak? Okay, so of course your first assumption is, well, come on, it's obviously the DNA primary sequence. Whatever it is, it has to be that. So what and then with Aviv or Geoff's approach and promoters with massively, I use the word much better, massively, but massively parallel assays in which he created many more variants of the sequence that could ever exist in nature to start giving machine learning approaches so you could start understanding. And then Mike Levine's famous paper where he showed some examples in a fish model that basically everything matters: spacing, strength of sites, et cetera, et cetera. So, of course, the answer is we don't know the grammar, and we still don't even know all promoter grammar. So, therefore, it's a very large quest. We're working on it. We, as graduate students, Stephen Gann, and someone's been in the lab for a time, very fortunate, Amir Gamilel. And basically, I won't give you even our strategy, but what we've learned so far is something that's, I guess maybe it was obvious from the beginning. The strong enhancers are the ones were basically the most basally inactive. You want the big impact of a signal-dependent transcription to do something, you want it to bind and do something. And that's exactly what happens. So they're the most inactive it's not they become stronger than the other enhancers, they just become stronger than they were basally, and they have the most impact. And because of a strategy licensed by the ENCODE project, which has now on certain cell types gone through the work of tagging the endogenous transcript of now 850 DNA binding transcription factors, so we're hitting up there now, and knowing the cut and tag or cut and run or ChIP-seq outcome. Now we can sort of start assembling, at least to that accuracy, which factors are not predicted, now actually bind to the enhancers at question, and that analysis is way over our heads. We're getting help from a very good informatics group who's now moved to China. And the answer is we're not there yet at all. But I can certainly tell you that the ones that are strong enhancers, interestingly, have the least number of the extra factors bound. And there will be some factors, however, that are not bound. And the weak ones are the ones that have more factors bound, although there's some that will not be bound. And they have a higher basal activity. So the big difference between the ones that are strongly enhanced and those that are least enhanced, at least at a superficial level, is that the strongly induced will have the most quiescent state in the basal with the least number of additional transcription factors. And the ones that are weakly were already somewhat better. They will give less of a differential when estrogen receptor did the same. Now, then you can ask me, well, what about the estrogen binding sites? What's the difference? And there the mystery starts really more complex. Because the estrogen binding site, because it's a canonical site, and everybody knows now, and now it's very beautiful data, that nature doesn't like strong binding sites on regulated systems. Never. Estrogen receptor, back to Valentina's day, we always were struck that the things that have been done by biochemical assay, those sites were never in the genome. They were weaker sites. So why did you never use strong sites? And now we know. Because if you make weak sites in some system, such as limb development, into strong sites, you get extra limbs and extra digits, or etc. So in other words, the systems depends on weak binding sites and ways to perturb that site, and that's true here. But even when we use the word weak binding site, we look at the strongest enhancers and we say, well, there's a canonical, 5 base rate recognition, 3 intermediate, and an opposing direction, essentially core binding motif on the other side. That's the ERE, estrogen response element. And for 37% of them, we see it. Well, what about the other huge percent? Well, if we then only count a half site, we can get up another 30 to 40%. So now we're getting into the 80-some%. We don't have them all. So a lot of them, by our current definition of a site, are essentially, they don't have a site. Now that's because we still don't know the real sites for any transcription factor, but estrogen receptor, one of the better studies, still don't have it. Okay, so why are these sites silent? Who knows? But obviously it doesn't take much imagination to think, they must be marked, they must be a code. And of course, the beautiful work that's been done on the methylation, demethylation cycle by the TET system is really so pertinent here. You'll notice that a lot of bases are capable of being methylated in the classical way, but once the TET system goes to work, and we end up finally with just a 5 CAC mark, now the number of sites in the genome, but we have a problem. Methodology is terrible, and therefore no one really has a true number, but it seems that everyone agrees it goes down to a very small percent of the sites. And where are those sites? at least as best we can tell, most of them are enhancers. So in other words, now we can say a strong enhancer is basically inactive because it's marked in a particular way. So the hypothesis, and that's all it is, the hypothesis is that you need a way probably to activate the estrogen receptor must cause a change in this mark and a start of the system. So this is where, again, Yunnan Tang has done a tremendous amount of work, and I just have to tell you, of course, doing all kinds of biochemistry and mass spec, I mean, it didn't escape our notice that interactions included TOP1 on these enhancers. And here's our mark, 5-CAC, by ineffective methodology and relationship to ATAC-seq data. So the more open you are, the less the mark. The more open, the less open you are, the more mark. So in other words, again, a correlation that argues that is regulatory. And then we know that, interestingly, nuclear receptors like to interact with this enzyme that actually makes the demethylation occur, which is TDG. So TDG is brought in with the estrogen receptor itself when it binds to DNA. Okay, and where is TDG? Now that, luckily, we can do cut and tag, but we learned the hard way, cut and run is very great for enhancers. Don't use it on promoters, big mistake that we all make, and don't catch for years, and then we figure it out. And it's mostly on regions that we would think would be enhancer-like. And we can do a knockdown of TDG. And we look at on volcano plots, we can certainly see that a large number of the inactive and known inactive program goes away. So in other words, not only do we bring TDG and have the capacity to demethylate, we also affect the program dramatically. Okay, so the next question is, the next step is, so if we knock down TDG and we just look for topoisomerase 1 that's covalently linked, that's so-called TIL-CC, do you need this TDG to get it to work? Yes, you do. So in other words, inference, the target demethylation program must be very important. Here the summary of the active strong enhancers, but what we are missing is the actual measurement of 5-CAC on the enhancers. Why are we missing it? Because the methods don't work. So we've had to work, we're working on a method. We're not there. We can now get about 35 to 40% of the sites. We don't have them all. We're working on it. It's really, well, the word working on it is like a year. So we're trying to get there. Okay, so assuming that last step is true, okay, and that now asking you to go along with me, here's what we're thinking. Okay, in the basal state, the strongest enhancers are decorated with 5-CAC. That is a unproven statement yet, because I can't say that they're all decorated with 5-CAC. Okay, now we give the estrogen receptor binding, and now I've already shown you the estrogen receptor can bring with it not only the kinase that it always brings, but the other thing it brings this TDG. Now, because of the fly clue, we knew TDG could interact with topo-CC, which we checked. Yes, true, it does. And apparently, our data would suggest it activates the topo-CC. So the first mystery, why did estrogen cause topoisomerase to get activated? Probably because it basically allows not only the demethylation of the region, but in a way that we don't quite understand. It activates topoisomerase and you make the covalent link. And we already know once you make the covalent link, then you bring in Q70, then you bring in phosphorylated form of HB1 gamma at 26. You can elongate, you can make a good enhancer out of what was before an open but not effective enhancer. Okay, so sorry, I had a technical glitch. With age, this is not a picture of the real curve, but this is a schematic that says the older we get, the less TDG we have. So I'm speculating and interested in the idea that aging, amongst other things, is a compromise of very important regulated transcriptional vectors in the brain. For example, since depolarization uses the same rules, this would probably mean you really don't. Learning memory is down. Gee, that's a fact that people observe, and so on and so forth. It's probably accelerated by things that we call genetic differences that change the rate at which we have changes and loss of mental acuity. Okay, so the hypothesis then is signal-induced enhancer activation requires DNA demethylation of 5-CAC marks based on regulated TF-dependent recruitment of TDG. So that's where we are on that step. Now, it would take me a whole seminar to present the next part, so I'm going to give you the summary statement. 2 people in the lab really took 5 years of their time to set up the real-time imaging and on single loci with all the tags so you can measure the RNA expression, the DNA, where it is, we can do the motility, and then we can mix that with single molecule imaging, and we can try and get some idea of what's going on. And the summary statement is, when you're not activated by estrogen receptor, you're not very motile in that area. When you give estrogen receptor, you do get more motility. But interestingly enough, Michael Levine, again, came up with this beautiful demonstration of flies of what he calls bursting, which is now clearly the case. So you give a signal, the gene goes on, usually a lag of time, and the gene goes on, but it doesn't stay on, it goes back off, and then at a certain random periodicity, but not so random in flies, you then come back on. So now the fly is beautiful, you can do all the stripes, you can do it in the whole set of things. So the amount of data and statistics is gorgeous. When you do it in a mammalian cell, when you have one locus, the statistics is not gorgeous, and it doesn't give you a global picture, but nevertheless, you can get through data. I'm not going to show you, I give you 20 slides on this, and that would be the end. So I'll just say, of course, you can recruit the bursting of your gene. You give estrogen, 10 minutes later, blah, 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 you get it on, it stays on for variable times, 10 minutes, 20 minutes, goes back off, then goes on. One of the essence of life that I think is fascinating and still, as far as I know, unsolved, is the asymmetry of cellular response. I mean, from yeast to man, if you give a signal or you give a toxic insult, all the cells don't die, all the cells don't respond. Only a subset respond, and that is probably the key to survival, that you don't get killed off by one event, because your cells are asymmetry in what they're capable to do at that moment in time. Okay, so they burst, and you look at it, there's a statistics, and early you get more bursting, later you still get bursting, but about around 2 hours in, the amount of bursting rate goes down at least 50%. And that's what we call, but maybe we shouldn't have coined the term, acute versus chronic. I think it's very common in almost all systems. You know, if the signal persists, you don't have as robust an effect, and it's a great question, what makes it be robust early and not robust late? Okay, so I won't beat this to death. And finally, couple this to single molecule imaging. I'm not going to go through the whole shtick on the data. But then you start finding many states, because now it's just crude. Now you're using microscopy, you're just trying to say, what we see, oh, there's a population, counts about 8% of all the estrogen clustering we see in the cell, in the nucleus, and that 8% is the one that's, I'm sorry for the detail, 1,6-hexane-diol sensitive. In other words, it probably is in some kind of condensate, just like we think is on the strong enhancers. So we think these are the strong enhancers. These are 1,6-hexane-diol sensitive, and that group is what we think represents the regulated enhancer population. And from that data, and doing a lot more work, including GMM and agglomerative clustering, we can say that there's a difference between the bursting and non-bursting. When you're bursting, that is, your gene is on, you're more constrained than you are in the post-burst. So you must make something about the topography, the condensate, change during the time when you're really actively transcribing versus not. And of course, I can't answer why that is, and no one can at this point because their methodology is just there. But with super resolution microscopy and new techniques, and a group in the Netherlands has come up with a microscopy arbitrary approach. They think tries to show by movement difference between bursting and non-bursting. So let's just say that I think the model would be, why would you constrain? And I'm going to get back to what I think it is. Okay, then finally we're down to that step about the so-called checkpoint. Now this has been a lot of fun. I really shouldn't have, I sort of browbeat two very talented postdocs in the lab and joined the lab, Lixuan Wang and Wei Wan, to take on this old thing because we had gotten interested in a famous thing called on promoters it's called promoter pausing or promoter pause release. So the promoter has polymerases there, you're initiated, but you don't elongate. And everybody knows that's all happens on promoters, but somebody who actually is a cohort with thing of Wen Liu, who's now a dean of a school in China, went on to show that enhancers could have this same machinery that's called the PTFB machinery. In case you're not familiar, so PTFB is why you can elongate, and it has a huge complex it makes, and it's held together by our little RNA that's non-coding called 7SK, an ancient retrotransposon. And that 7SK is capped, and he showed remarkably that if you decap the JFJD6 an enzyme would decap that, and you release this, and you need another protein called CAP1 to keep everything off. And then if you go on, you turn on the kinase, part of the PTFB complex. I guess it's not demonstrated well in the way it's drawn. And as a result, you can get the correct phosphorylation of polymerase, and you can elongate. Okay, so what they questioned, that I stuck with this, do these events occur on enhancers? After all, that's the first transcription unit in our mind that starts the process, so it should happen on enhancers. And in 3 years, through actually the initial review process, they got the answer. And the answer is yes, of course it does. It's the exact same thing, it's just very hard to measure. These are shorter transcripts, blah, blah, blah. Anyway, you do have the same players. What makes this system work is and this had been previously done by someone who's in the lab, who's now back in China also, who had found that CAP1 does get on the promoters in the inactive state, but on the enhancer, we found here, they found that it actually comes on only when it gets recruited by being phosphorylated in a specific residue. And that residue of CAP1, when it's phosphorylated, so it's brought in by its ability to do this, is that kinase that come in with the estrogen receptor again, phosphorylates CAP1. And when that happens, phospho-CAP1 loses one of its functions, which I didn't even realize had, is a sumo function, sumo ligase. So the sumulation of the kinase component of the PTFB is actually now altered. And so now, what was constrained before, so its kinase was not active, becomes active, and now you can make the change in the polymerase, and you can elongate, and you can go on. And you fold apart. Did you have the inactive form in 7SK? Yes, we did. It's there. So it's on the enhancers, everything is there. It's very hard to measure elongation on enhancers. Some of them are only hundreds of base pairs, and we had to do some lot of tricks, but anyway, now it's very clear. There is a pause release event on enhancers, and so we'll call that a checkpoint. So now we got through the checkpoint. Quantitatively important? Very. So in other words, each of these 5 steps, if you didn't know about them, you would not realize that we're actually, truly, every one of them is a checkpoint. Okay, so now we can get back to the same curve, and we get finally to, thank goodness for you guys, the last step of this process, which is the project that I'm very surprised by, and that is about a region of the estrogen receptor called the intrinsically disordered region, IDRs. Every transcription factor, of course, most proteins have large IDR regions. They have structured regions, the binding region, the ligand binding domain, etc. But on the other side, the N-terminus, not so. And why do you need the IDR, or do you need it? So here's the question. Nuclear receptor paraspeckle condensates license acute enhancer activation. And this is the work done by a fearless graduate postdoc that came from Tsinghua to the lab, who had worked on mouse work, had no knowledge of transcription or molecular biology to that matter, and really took 2 years to get to speed, and then has really taken over to do, really, I think, a lovely story. And the story, of course, is that the estrogen receptor, back when Valentina was making key strides in this, had been described already as having domains, and the first one was called activation function 1, which meant that if you took it off, the estrogen receptor didn't work as well, and nobody had any idea. And then there's the famous ligand binding domain with the hydrophobic pocket as charge clamp regulated for the so-called LXXLL coactivators. And so they knew why this was called activation function. There were a lot of people did the crude biochemistry and this and that, and they said, oh, I think this is a coactivator, and it was all pretty much not right. And so basically, it stayed clear, we didn't know why this huge disordered domain was so, well, was it important at all? So was it important? The answer is, if you took off those 170 or 150 bases, or somebody said, oh, well, wait, that's 150 amino acids, that doesn't mean anything. You make the finer mutation. So we made 10 base substitutions and 2 different amino acids, and yes, same result. So you do need the IDR. We certainly don't know what the induced fit story is, but we certainly know you need it to bind well. Now that was a shock, because, you know, by prediction, this had nothing to do with DNA binding. So why in the world would it change the binding? The look is pretty dramatic. Get rid of the IDR, it doesn't bind well, and the next question is, really? If you do a biochemical assay like an MSA, do you get the same result? The answer is no, you don't. You end up, it's perfectly fine. So in the cell, it doesn't work. In the real world of biochemistry, it does work. What happened? And now I'm going to spare you the tons of slides it would take to go through this. He did something very smart. He created a molecule, an estrogen receptor, which he mutated the so-called P-box, which means it can't bind DNA effectively. Then we found an old paper, which we had completely missed, very minor paper, that somebody said, oh, I found an amino acid that the estrogen receptor doesn't dimerize. He said, oh, sure. He was right. One amino acid change, you can see terminus. So now we have something that can't dimerize, it can't bind DNA. So of course it should do nothing. And it by itself, of course, the answer is it didn't do nothing. The answer was, and I won't go through all the experiments, if you hook it up on a site that has a normal estrogen receptor binding, and it has the IDR, and that has the IDR, this guy, nevertheless, can be found to get recruited to the estrogen-induced strong enhancers. So much so that it's very compromised, and he was able to do the next step, which is to, by some tricky things, to argue what percent of all the estrogen receptor on an enhancer that we call a strong enhancer is actually on DNA at a given moment in time. And the answer is about 30%. And the rest of it's not on DNA. It's in the condensate. So most of what we call a strong site has some estrogen receptor bound, and then some that's not bound. Now, I learned something today in the discussion about on rates and off rates that is very striking to me. So if you read most papers, they describe, okay, what makes something have a strong site? And they all say it's retention time. If you retain it a long time, then it's a strong site. It's weak site. So I don't think it's that simple because of the following. Again, cartoon. So notice we got nowhere into this story. A long time after many failures, we said, okay, come on, let's take advantage, I'm sorry for this cutoff, for Alice Ting's beautiful work, proximity labeling. So take 6 months to make it work for your system and blah, blah, blah. And of course we can find that if we test your receptor, and we can certainly see all the players that we knew were going to be there that are in the megatrans complex. But there were 4 proteins that were very strong that always showed up: NONO, SFPQ, BSPC1, and RBM14. What are they? They're turned out to be components of a structure that I knew nothing about called the paraspeckle. What is the paraspeckle? Okay. Well, of course, I've always been amazed by the cell biology field, which I know nothing about. George Bellotti was an inspiration in that field to not figure out all these structures in the cytoplasm and what they did. But the nucleus, even the Cajal found the first much more than 100 years ago now, even though we know something about most of these structures, we don't know that much, really. But we know they must be important in their structures, and a lot of them are known, at least as well as people can tell, to be phase-separated structures. They're memberless structures, but they are held together. And one of them is really the one I knew nothing about, because most cells don't even have a visible paraspeckle. So the paraspeckle is often seen as a big blob next to the nuclear speckle, or it's also called the interchromatin granule. We had already known, and we knew from Cold Spring Harbor work, that the interchromatin granules are linked to many splicing and transcriptional coactivators, and are presumed to have a transcriptional role. The paraspeckle, no, but it does have splicing factors in it. And what is a paraspeckle? A paraspeckle contains a very famous long non-coding RNA called NEAT1. It's a very abundant RNA, so abundant it's almost whatever you do, you can't get rid of more than 90% of it. We just spent 2 years of trying to get it better than that. What many labs found is that it assembles a group of proteins, starting with this one called NONO and SFPQ, across the length of the RNA, and then they cause conformational changes so that you end up with this core of the paraspeckle. So 2 lessons I've learned from just looking at this. Wow. So in other words, when we say a protein dimerizes or heterodimerizes, let's not get too proud of ourselves, because if there's an RNA involved, they don't actually have to have very much interaction affinity at all. They can just bind the RNA, and it sort of assembles what we call a homodimerization or multimerization. So the word multimer, and I'm going to end with that story because Valentin wanted me to, and you guys can stand it. So that's how you make the paraspeckle. So the little problem, if you look at most cells, we can't see any. When you do see them, there are about 6 in a cell. So now, how dare I tell you the next thing? So the next thing is that knockdown of the components, like NONO, have a massive effect on binding and function. So in other words, our cell, where we don't see any paraspeckles, this machinery that we said is linked seems to be functionally linked. So why is that? And again, I'm going to spare you the next 15 slides it would take to just say, well, the IDR region must be it. And now we found what the IDR region seems to interact with by this proximity labeling. It seems to be this NONO complex. And does it really interact? Yeah, it really does interact biochemically. And NONO is functionally required. And I didn't put in the slide, but we finally got it. and the NOMO distribution in the genome, sure enough, is on all of our strong enhancers. So in other words, everything fits except the logic. And that's the problem. Okay, so the next thing, again, cartoon. So he did a series of experiments, and they're absolutely complicated experiments that mean there could be interpretation problems. The idea in this was, let's take advantage of this, these mutants that can't bind DNA and can't dimerize, and let's see if A, estrogen receptor has to be bound to DNA and has to have its IDR for it to do anything. Answer, yes. Second, and shocking thing to me, and I still want to check this, is that I don't understand this. So when we started, Valentin and I, on estrogen receptor, we read the beautiful stories that talk, estrogen receptors in the cytoplasm, it's on heat shock proteins, and you give estrogen, that's association loss, and it goes to the nucleus, and it goes to its cognate sites. Revision by many people, no, you're completely wrong. It's largely in the nucleus all the time anyway, so that's not the whole story. But the answer is, you still need estrogen receptor. Well, that brings the question, does this paraspeckle interaction, well, again, dependent, and I really want to get another way to do this, but our data right now say yes. So in other words, instead of just floating around in the nucleus, it then finds a place to spend a small amount of time. You know how these are. These are dynamic stories. That means it spends almost no time on the paraspeckle, but it's more likely to be on a paraspeckle than anywhere else. And it's just like TAD boundaries. You look at any given cell, it doesn't have the boundary, but you do the informatics on all the cells, then you can see the TAD boundaries, right? So the same thing here, that statistically, it's a loading zone. It's a concentrating zone. Now, did this need the IDR to get to here? Yes. Did it do anything? Answer, yes. If you do the estrogen receptor, and the long story short, so in order to do the experiment, you have to knock down estrogen receptors to only 5%. Now you only regulate 56 enhancers are strong. But those 56 all need this machinery. And this guy, when it has the IDR, can make it stronger. So it is contributing transcriptional machinery to get into the condensate. What machinery? Well, Luke, we can tell you better than anybody, and Val can tell you better than anybody. Of course, there was the LXXLs, and so we made a 1 amino acid mutation that would eliminate the charge clamp that lets any LXXL coactivator, which there are many. We've never understood, why do you have so many coactivators that all bind to the same site? Well, it's probably stochastic. And the answer probably is that by having a huge number coming together, now you have a big repertoire of coactivators that all condensate, but they're not on DNA at that moment. But what's the off rate? And now I realize how shaky the original data was, but the general is in 5 seconds of photo-bleaching, you restore. So within seconds, whatever's on DNA, which was supposed to be very stable, was not really supposed, it had enough time to come off and come back on in seconds. So the way we're seeing it is, it must be an on-rate determinant. It must be that by having all these condensates brought by the paraspeckle, that the next time on, you have a much higher concentration. And if you mutate, the ability to also bind LXXLL coactivators, now it doesn't have a transcriptional effect. So in other words, it's bringing the power of more robust transcriptional activation. So that's our model, but obviously we'd like to end with a beautiful photomicrographic showing that the paraspeckle is with this and it's sloppy. So instead, we generated an AI to generate a picture of this. And so here's the model it comes up with, that basically we need now super resolution microscopy, that's what we're trying to close on, to show that there is not a paraspeckle. It's a mini paraspeckle. It's been missed in my cell biology up to now because of technology, and that actually that we think it's the same is true for the speckles and everything else, that there are really hundreds to thousands of these, and that really it's a dynamic kinetics that gets you to be activated. And that's the punchline of this. So basically the N-terminus locates CR to the paraspeckle, and required for ligand-dependent enhancer binding. It permits the condensate without many paraspeckles on being poetic license and eRNA enhances them, and stochastically it brings in a lot of LXA cell coactivators. And does it apply to other enhancers? So we didn't get clean results on the AR because of technical reasons. We're doing the NF-kappa B inflammatory system right now, which is very promising. And normally I was going to end the thing there, but I had one thing depending on my
I think we are running late.
We're out, so we're going to
Yeah.
call it here. So I'm going to just say, so those are the 5 phases. And I want to just say that Yulang Tang, really, I thought this topo-1 isomerase part of the story is amazing. Susan and Tom spent the 5 years of getting the single cell real-time imaging. Likun, thank you so much, Likun, for doing that amazing story on the paraspeckle with a lot of input from Amir. Lixuan, Wang, and Wei Wang got the PTFB checkpoint done, and I think, since I'm not going to tell you the last story, then thank you very much for letting me have this little too fast story on enhancer activation.
Thank you so much. Yes.
So thank you for this very exhaustive.
Exhausting. Yeah,
Well, I want to just touch on one thing that you didn't mention. What's the role, if any, of the enhancer RNA? Is a byproduct of the enhancer activity, or in some enhancer has a
This is the question that's haunted this finding since the first report. First of all, no one believed there was transcription. Now everybody believes there's transcription. Now, do all the enhancers show the eRNA? And the answer is, of course, technically, it's very difficult to get a good number when you do single nucleus, etc. But the answer is, in my opinion, yes, always. I don't believe those who said you don't get it. You just go below statistical significance. I think you get some enhancer. Do you need, is it the active transcription, or do you need the RNA? Well, I don't think my answer should count because I'm biased. I think the answer is yes, you do need the RNA, but why you need the RNA is another question. In my opinion, there's a lot of studies with ASOs and everything else that all have technical concerns that yes, you need the RNA. It's not just the active transcription, but exactly why, at a really nice molecular level, I think would be hard pressed to make too much of a case. It does increase the condensate. It does increase the recruitment of various things you see, including cohesin and so on. But do I believe that that's completely the story? In my mind, yes. And another story that I didn't have time to tell, I can show other examples where nearby RNAs that have no specificity whatsoever play a role in enhancer grammar, let's just say that, and promoter grammar, because of a concept that I'm just searching for a name, which we call RNA equipoising. And the RNA equipoising says that you have a very, in 1974, there was a paper that was a mathematical paper that tried to say, if you have 3 things, including it could be an RNA, 2 proteins, and you get a curve of that binding, it will always be a biphasic curve. They go up and they go down. If you have too little RNA, it doesn't work. If you have the right amount, the Goldilocks effect, it works. Go high, you don't even get the condensate or the interaction. Now I can tell you that biologically is the key, for example, why CTCF works on promoter cohorts and so on. So do I believe that is true for enhancer RNAs? Of course I do. And therefore, we don't need a ton of RNA to have a very important biological function to get the Goldilocks effects of getting things set up correctly. But, doing the experiments to really prove this in a absolutely ambiguous way harrowing.
Steve.
Great talk, thank you. Questions to do with the relationship between the enhancer and promoters. The first question being, how many promoters are regulated by an individual?
Okay, so this is such a great, fun question because so much time is spent arguing about such a minor point. So it's amazing. You know, we all know it. You know, within a TAD boundary, you can see it as, you know, the neighborhood. Of course, every enhancer is going to go visit everybody in the neighborhood, and it's a matter of time spent, which used to be called retention. I don't know if that's right, and loop extrusion and all this, and how much time you spend in each boundary. But the answer is, you could enhance probably almost all the promoters there, and probably do to some small extent. In our examples, you do to some small extent, but it chooses one. If that promoter doesn't work, it chooses the next one to be its target. And if that one doesn't work, it chooses, and what was the difference? How much CTCF is bound on those promoters? So, so I guess in answer to your question, I think the answer is, of course you can activate more than one. Now we know examples of ones that can do it hundreds of kilobases away, and that's because you get spatial proximity of these very distant regions, and we won't do into why. And the only methodology that's really making this convincing is this, to me, is, well, there are lots of them that are used. They're the variants of high city. But basically, I think the Marefish data is gorgeous. And to me, it's very convincing that if you interact at a statistical level, you'll spend more time near the other place you're regulating. And that's the model is called loop on loop.
That's sort of a continuation of this is location. Within, and what I've sort of been thinking about for many years, based on lots and lots of other people's data, obviously, is location from the point of view of the role of the nuclear lamina itself.
Oh, now you're getting to a very wonderful. That's another issue. That's a great question. You know, yes, how much dynamics is there in where you are, and that's why that's why I'm so intrigued about this motility issue and bursting, non-bursting. It's very interesting. So of course you're absolutely right. It's what you spend time with. That gives you the cofactor machinery, but where the looping machinery, on the other hand, is intrinsic and local. But we think of distances between chromosomes. I mean, now it's very clear from a lot of data, although many people would say it's still not unambiguous. If you're highly transcribed as a boundary, about the 20% of boundaries that are very high transcribed can interact with a certain number of boundaries on many other chromosomes. By interact, we mean have a greater probability of spending some time together. Very minimal time. So you wouldn't just see it in microscopy. But you definitely do spend more time, and those guys had as a feature the highest transcription rate. So now a lot of people are thinking, I'm thinking, if you have a very highly transcribed enhancer RNA, you have a non-coding RNA. And that non-coding RNA is going to essentially help cement these very long distance interactions between chromosomes, so they must spend more time. And now we're getting into this whole idea of how you distribute factors in the nucleus. And of course you must be right. And the lamin, the nucleolus, et cetera, et cetera. So these are, and the weaker the affinity area, like an enhancer.
Actually, Geoff, thinking, combined to what Steve was asking, I'm really intrigued by your idea that the stronger enhancer are the more repressed ones.
Yes,
and whether there is a role for this thing that we've always considered co-repressors that now are showing activating a role.
Did they have more polycomb kind of things on before?
The HP1 binding H3K9 and all those things.
It's not data that I would like to stand on. It is yes. Yeah. Okay.
Thank you.
Yeah. Oh no.